Let's start. Welcome, welcome, academic weapons. Because you guys truly are academic weapons. You guys are joining at 10 a.m. on a Saturday. You guys could be at the beach. You guys could be anywhere, but you're here instead. So I really thank you guys for committing to this and joining, because you guys are the committed students that we are trying to help out as well. But a bit today in terms of who am I? You guys are probably wondering, like, who the heck is this guy on my screen right now? And why is he even helping me? Like, why is he helping me? What's the purpose here? Well, let me share my screen when I do it. Let me put my face cam while I do this so you guys can see me face to face. But hey everyone, my name is Jad. I graduated from Masterclass Study Hub in 2022, sorry. I got an ATAR of 99.6. And for the past 2 years, I've been trying to help people out, not just in biology, but just ATAR in general, giving you guys strategies more than just the content knowledge, but actually the strategy you can walk away with and ace every single exam you guys get for QCAA. So I spent ages trying to dissect what QCAA is actually asking for, because so many students are like, do I need to know this? Do I need to know this? How do I even answer this question? So we're going to dive into a lot of that today, and the art of answering biology questions, as I like to call it. And what motivated me to kind of hop on today? Well, like throughout school, like 2 years ago, I was exactly in your positions. I know exactly how it feels right now. Mock exams, you're stressed out, and then some of these mock exams are giving you a bit of a reality check, as I'm seeing with some of these students. So it's like, oh, like I didn't know that much. I'm cooked. But today we're going to try and heal your uncooked syndrome and try and transform those grades and make sure that you guys get the maximum ATAR that you can, because throughout school, all my mates was in the exact same situation. I was looking around and everyone just was struggling. But it doesn't have to be like that, guys. If you guys have a strategy moving forward and you guys have a framework for answering questions, it becomes a lot easier, which is what I'm going to try and cover today. For the most part, strategies, understanding the common questions that come up again and again with biology QCAA, and then the common student mistakes and misconceptions that are made that QCAA always tricks half the cohort on. So we're going to make sure you're not that half. You're the half that actually prevails on top, and that way you get on the end of the bell curve and your ATAR bumps up, because bio has a bit of a conception that like it's the one out of the sciences that usually tanks your ATAR because the scaling isn't as bad. Everyone and their mum does bio, right? So you guys have to be in the top notch to make sure that scaling is in your favor. So that's what we're going to go through today. All right. So just before I start as well, I just wanted to show you guys that what we're about to show you, what I'm about to cover today, has helped students that are exactly in your position. So if I just want to zoom into a couple of these reviews and stuff. So this is off Queensland Study Hub, but we've got students, like I told you, Thomas reached out to us for his mock exams. He had like 40% on his mocks. Through 3 weeks of sessions like these, he was able to get 80% in his final external exam. He doubled his results, which is crazy. We've got students who were accepted into dentistry with 99 plus ATARs because of these master classes. We've had students that got in med because of this. So what I'm about to show you today is obviously as much value as I can give. So make sure, hopefully you guys can also see with these examples, hopefully some of you guys can see yourself in one of these, make sure you guys are paying attention, put those phones away. So would it be safe to say, or fair to say, that I can take 90 minutes of your time today? Comment on the chat. Everyone, drop a thumbs up if I can have 90 minutes of your attention today, because what I'm about to show you is actually going to change your view on QCAA Bio. Is this being recorded? Thumbs up. Yeah. Awesome, guys. Everyone get a thumbs up. I'm not starting this until you guys get a thumbs up. If it's a no and I don't get 90 minutes of time, you might as well leave now because we're only trying to help our students that are ready to transform their ATARs and to make sure that you guys get to the next level. And we should be acing everything to it. Awesome. Thumbs up. To answer your question, AHA7943, is this being recorded? Yes, this is being recorded. But the catch, or not the catch, but is we'll only be sending the resources of this class to students that stay around to the end and fill out a little survey. So be sure to stay out to the end, stay all the way to the end. Number one, obviously, to get all the value I'm about to give you, but also to fill in that little form that I'm going to give you guys, so we can have your email to send out these resources. So yeah, well, good guys. Awesome. So many thumbs up. Is this the only session you're doing before externals? No. So we're going to do a lot more. We're going to be going through more of these, because as you guys can see, and I'll start here, understanding the scope of biology. So for QCAA, look at how much learning goals we have, right? So obviously I can't cover all of this today, but by the end of externals, I will aim to cover all of it. This is just the first intro session. So if you guys like today, be sure to fill the form at the end, and I will make sure that I take you guys on this journey to getting 100% on these bio exams. And I'm not even exaggerating when I say 100%. You can get 100% because everything that is allowed to be assessed is here. They tell you. So if you guys haven't already, I'm going to get you to do something very powerful after this session, which is I'm going to give you guys access to this. Obviously, those that stay to the end, and you guys need to highlight. You guys need to highlight green, the ones you feel confident with, yellow, the ones you kind of like you seem all right with, and red, the ones that, well, this is purple, but you get the idea, the ones that you don't feel confident with at all. This is called the traffic light method. So powerful, especially for bio, because this is all QCAA assesses you on. Absolutely everything they assess you on is here. So there's two aspects to this. The cognitive verb is a big one, right? So you got to look at this, right? If it's recognize, right? What does this mean, guys? Recognize. This means you just have to appreciate that this exists, right? Recognize that biodiversity includes a diversity of species. So if you know that, that's that ticked. This is an instant green for you guys. Boom, you got one. You got like 100 and something to go. But you guys get the idea. So the ones I've kind of put a little asterisk on in blue, these are the ones we're going to cover today. And the reason I've chosen things like these is these are the things that students usually miss on the exams. And there's a lot more of these. Obviously, as you guys can see, there's a lot more than what I'm going to cover today, which is why we have future sessions. So if you guys like today, stay tuned for that. How are we all feeling in terms of looking at these learning goals? Let me check the chat real quick. How are we all? Are we getting overwhelmed looking at these learning goals? Are we feeling confident? How many of these do you reckon is red? Is your whole page red right now? Is your whole page green? I'm in the middle. Yeah. Anyone else? How are we all feeling looking at these learning goals right now? Are we kind of shocked of the sheer volume, or are we kind of like, okay, this is all right? Most of Unit 4 is definitely in red. Yeah. For a lot of students, it is. Unit 4 is harder than Unit 3, although, and I'll keep this clear, Unit 3 questions, the questions that the content as a top of understanding, Unit 3 is much easier than Unit 4. But the questions they give you for Unit 3 sometimes tend to be harder to compensate for that ease of concept. So DNA is green, Unit 4 is more difficult. Yeah, okay, absolutely. Without further ado, let's get straight to it. So, just also as well, since I am the library and the internet, I'm currently at 2 bars. If there's any time throughout the call that you guys lose track of my voice, or can no longer hear me, or can no longer see the screen, just drop it in the chat, and I will be sure to attend to that for you guys. All right. So let's start without further ado. All right. The art of answering bio questions. Okay. So diving into this is kind of, there's a key thing when looking at QCAA questions, especially for bio. And there's 2 core concepts in terms of the art of answering questions. Number 1, what is the cognitive verb? And I mentioned this earlier as well. The cognitive verb is describe, identify, etc. They all mean different things, which means they have a certain level of detail. If you're looking at the learning goal, right? This is here. So looking at the cognitive verb and the learning goals gives you the scope of what you have to know, right? However, it's also not just in the learning goals, but the exam questions. Okay? So I will kind of, and the second thing, before I dive into more detail on that, is mark counting. So you guys probably wonder, what the hell is mark counting? This is looking at a question. Before you write anything, look at the number of marks, right? And the general principle here is, if 1 mark, it's 1 idea, 1 dot point, concept, 1 thing, right? So 1 mark equals 1 dot point, which you guys should know by now. But a lot of students don't look at this. They just yap. They yap, right? But if you guys pause before you write the question and think about this, how many marks and predict what each mark is going to be allocated to, combining the cognitive verb with the marks, you will get 100% unless you physically just don't know the answer, right? But this eliminates the possibility of you knowing the answer but not saying what they don't want you to say, because that's a lot of things that I saw on the chat. You guys were concerned the fact that QCAA, it doesn't make it clear what you want you to write. This is how you overcome this: cognitive verb and mark counting. So just before I get into the cognitive verbs in more detail, because that's a big one, I want you guys to appreciate plurals. Watch for this, right? So if they say identify factors, what does that mean? More than one factor. So they don't want you to, if it's a 2-marker, this will also give it away. But what some students could do is they can say, appreciate factors, and they can write 1 factor, and then the second mark, they'll try to explain that factor. They'll identify the fact 1 factor, and then they'll try to explain the 1 factor. You will only get 1 mark for that. Why? Why? Okay, type on the chat. Why is that 1 mark? Two factors, exactly, because it's factors, it's plural. So because it's identify, right? That's just asking you to list 1 factor, list 2 factors. That's it. Not explain, right? There's so many students, and it seems stupid that I'm telling you guys this now, but you cannot believe how many students would just go off at a tangent and yap, right? They're like, oh, I know this one, and they start explaining. The question didn't ask you to, bro. Okay, so very, very important. Let's dive into these cognitive verbs real quick. So identify. I want you guys to literally just spam me in the chat. What's identify? What's identify?
Sorry guys, as you know, Jad's at a public library this morning because JCU's lost power, so someone might just be asking him something. Just bear with him for a second. Today's just been a bit of a rough morning at JCU.
Hello? Hey guys, I'm so sorry that happened. Hello, can you guys hear me now?
Yep, you're good.
Sorry guys, I cut out. I'm so sorry about that. I try to be fancy and join on my laptop, so I have like 2 screens to look at, and it just like muted. I'm sorry guys. Alright, hold on. I'll try and join back. Share the screen again. Alright, cool. Okay. So, can you guys see the screen all good now? Yep. Okay, sweet. Too easy, guys. Alright. So, identify. What I was saying, exactly right, guys. Just state what it is. So this is easy. Easy, easy, easy. 1 markers. 1 markers. Just say it, right? Okay. Now, what about explain and describe? Right? So let's start with describe. What's the difference between describe versus explain? If they ask you to describe something versus explain something, what is the difference? What is the difference, guys? Explain is going into detail. Yeah. You guys can hear me still, right? Someone texted on the chat that you still can't hear me. Yeah? Okay, you guys can't hear me? Okay, so EA4L. Maybe fix. I don't know what's going on with that guy. Yeah. Alright, so yeah. Explaining is going into detail. You don't know the difference. Yeah, that's normal because a lot of students don't know the difference between the two things, right? So, significance for explain, you're close. You're getting up. You guys are getting warmer. Right, let me kind of dive into this real quick because this is a common misconception. Describe versus explain. Describe is literally what is going on, right? Like what is actually like describe what's happening, right? So like it's a process. It's more like you describe it, right? So what is going on, and also you want to say like characteristics and features of whatever's going on, right? Whereas explain, this is you want to dissect it in more detail, right, and bring in other factors. So essentially, explaining is giving reasons for it. That's the key differentiating factor here. Giving reasons, right? Whereas describe is just what's going on. Right? So explain is actually the more detailed version of describe. Okay? Alright. Now, I want to get to the big dog here. This is a big dog analysis. Let me write this down. Big dog. Right? Why is this a big dog? Well, I just got this from QCAA. This is QCAA's definition of analyze. Look at this. Just look at that. So let's dissect this really. Dissect to a certain and examine constituent parts and all their relationships. Break down or examine in order to identify essential elements, features. Examine or consider something in order to explain and interpret it. The purpose of finding meaning, relationships, identifying patterns, similarities, and differences. Okay, that's a mouthful, right? So what does this actually mean? Well, I'm going to tell you guys very simply. It is pretty much going even deeper than explain, and usually this is with data. So this has to come with context, right? And you want to say relationships between things. So what is this? Relationships, similarities and differences, and then also trying to break down and explain the features. Okay? So as the big goal, whenever you see analyze, this is you want to pour your soul into this question. Okay? Does that sort of make sense, guys? Analyze? Analyze makes sense as well? Yeah? Okay. Alright. Now, compare versus contrast. This is a big one. Can you guys tell me what is the difference between compare and contrast? What are they even talking about, bro? Compare and contrast. And I'm going to show you guys common compare questions and then common contrast questions. So you guys can actually expose yourself to questions as well. That's a big part. Compare is similarity and differences. Contrast is differences. Yeah, okay. Alright. You guys are really good at this. Very nice. Okay, what about you're kind of missing something for compare though. It's similarities, differences, and what's the third thing? It's not just similarities and differences. Significance. Nice, Trisha, right? Significance of the difference. You guys are probably wondering, like, what's significance mean? Right? What does significance even mean? Let's go into significance is essentially relating this to the real world context or the context in which you say the difference. For example, common classic compare question. Compare oogenesis with spermatogenesis, right? So the difference, you can say, oh, there's like 4 haploids here versus one haploid with 3 polar bodies, redundant polar bodies. That's a difference. Comparison is it forms genetically diverse haploids with half the genetic material, right? What's the significance of this difference? The one that one produces 1 and the other produces 4. It's because you can say something like, so essentially significance is saying why that difference is relevant. So why the fact that this produces 1 polar body and this produces, 1 haploid and this produces 4 viable haploids is because there needs to be a higher volume of sperm in order to fertilize an egg successfully, whereas the egg doesn't have to withstand harsh environments of the uterus, and therefore not needed as much viable gametes, right? That's the significance of that difference. Do you guys understand significance now? So this is another classic compare question. Compare micro and macro evolution. And also, I just wanted to bring up, since we're doing the art of answering Bible questions, what's the second part besides the cognitive verb? Marker counting. So this is a 3 marker. a 3-marker. So it's compare. We should know what these 3 things they're asking for are, right? We know what the 3 things, right? What do we say compare was? A similarity? A difference? And then a significance. Right? Makes sense. So we've counted the marks. We've looked at the cognitive verb. You should not get this wrong unless you physically just don't know, right? So what is actually the difference between micro and macroevolution? Can anyone try and give it a guess? What's the difference? Macro is large scale. Yep. Anyone else? No new species formed. Big one. Exactly. Yes. Perfect, guys. So macroevolution results in the formation of a species, which is over a larger scale, over a long time scale. Whereas microevolution is mutations, genetic drift, and gene flow accumulated over time. But it does not result in a new species being formed. Yes, exactly. So that's the difference here, right? The similarity is the fact they're both caused by mutations, gene flow, and genetic drift. They're both caused by aspects that change the allele frequency in a population, right? And the significance of the difference is, so the significance of that is that this is a common term we use, but the descendant is of the same taxonomic group as the ancestor. This is for microevolution. Obviously, macroevolution is a different. The descendant is of a different taxonomic group than the ancestor. Descendant meaning, if we're looking at a cladogram, right? Descendant is whatever is the target point you're looking at, and ancestor is where they came from. Right? So if it's changing over time, the descendant ancestor, if that is of the same group, yes, this is microevolution. Whereas if they have formed a new species, this is macroevolution. Does that make sense, guys? Yep. Makes sense? Okay. Awesome. Alright, now, getting to a contrast question. This is a classic contrast question. Whenever we look at contrast, we said, what was this? Just differences, right? Just difference. So if we're trying to find just differences, this is usually looking at numbers, difference of numbers or values, right? So they rarely ever ask a contrast in terms of a concept. So notice compare, that's to do with concepts. Contrast is usually to do with values and numbers. So how do we find a difference of values and numbers? There's 2 aspects, usually. You say, which one is larger? So you describe the difference. Describe the difference. And then you quantify the difference. Key thing. Whenever they ask you to contrast things that are values, you have to give a number. You have to actually compare the numbers. How much is A greater than B, right? Not just A is greater than B, but how much, the difference. Key thing. So many students just forget to say the number. Don't be that guy, right? So, example here. Let's use our art of answering biology questions to look at part A. Contrast, okay, boom, cognitive verb, contrast. The efficiency of the biomass transfers between each level of the pyramid. Okay, alright, let's start with contrast. We said it's the differences. Now, we're going to do some mark counting here. 3 marks. Okay, that means they're looking for 3 things. So instantly in your head should be like 1, 2, 3. This is planning time. Well, not planning, obviously proves that you can't write on the paper. But you want to think this in your head. There's 3 aspects. Can anyone tell me the first aspect? Who's the brave person who's going to try and tell me the brave aspect? The first aspect. Efficiency of primary. Okay. Reduces the primary percentage. Exactly. Okay, what about the second markers? What's the second mark coming from? Primary to secondary. Amazing. Okay, and the third mark? What's the third mark? Differences in each percentage. You guys are locked in right now. I love it. Awesome. Good stuff. Okay. That's it. That's exactly it. Right? I'm not even going to bother answering this. You guys got this. Right? That's contrast. All right. Now, let's go into a new cognitive verb here. All right. Explain. All right. Let's try this. Explain the difference in biomass transfer efficiency identified in Part A. Now, what are the two aspects of the art of answering bio questions? Number one, cognitive verb. We look and we see it explain. What is explaining again? What is explaining? Give a reason? Yes, exactly. Give a reason. Okay. Dissect in more detail. Perfect. All right. And then how many marks is here? 2. Okay. 2 marks. All right. So what are the 2 points they need here? Reason that energy is lost. Okay. 10% rule. Yeah. Identify reason and why. Yeah. That is literally it. Identify reason and why. Perfect. That's the 2 marks. So I would probably say if it's explain the difference, the first mark would probably be what is the difference, and the second mark will be why the difference occurred. Give a reason why. So yeah, exactly. So what can the difference be from? Right. So if you were to actually calculate this question, you would see that the energy transfer from producers to primary consumers is more efficient than from primary to secondary. So you would say that. That's 1 mark. The second mark would be why that exists, and exactly right, guys. This could be due to respiration. It could be due to heat transfer loss or things like that. The reasons that energy can be lost across the pyramids, right? You guys understand now how to answer this question? Anyone have any questions regarding these two before I move on? So this is just quickly an exercise to identify how to actually answer things in relation to their cognitive verbs. And I'll dive deeper now to specific concepts that we can go through. But does everyone make sense of that to start off with? Yep. 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 All good. All good. All right. Awesome. Okay. Alrighty. Alright, bear with me, guys. I'm just going through to. Alright, can you guys still see the screen?
Nope.
No? Okay. Hold up. I will try and share it again. Alright. Sweet. Alright, can you guys all see the screen? Yep. Can you guys all see the screen? Yep. Perfect. Okay. Awesome. Alrighty. So, let me get into now, specifically in terms of this learning goal here, population ecology, right? So now let's dive into population ecology. So if I'm trying to look through the learning goals here, right? I've done the traffic light method for you guys, in terms of what I think is the easy stuff and what I think is stuff that students usually miss. But if we look at the green stuff here, define carrying capacity. That is free. That is free, guys, right? Calculate population growth change. That is just using a simple formula. Linkler index. That is mark, capture, recapture, right? These are all green stuff, I think. It's just a matter of learning it. Now, if we're going to just to make sure we're all okay, can everyone give a go at this question here to try and define carrying capacity? So this is obviously addressing the first learning goal. Yep, B. Awesome. Can you guys identify why it's not D? Why is it not D? Because D could be another one that people could have picked. Different species, exactly. Yeah, different species. Awesome. Okay, now the ones I'm going to cover today is the ones I've said, number 1 and number 2. These are the key ones these form the big dog questions that QCAA asks, where it's like multiple marks and things of that sort. So the first one, explain why the carrying capacity of a population is determined by limiting factors, right? So there's biotic and abiotic. Explain. So this is give reasons, right? So just to kind of address this, what are things that can affect the carrying capacity of a population? Can you guys just list them out for me? What are things that can impact the carrying capacity of a population? Food availability. Yeah, awesome. Good stuff. Abiotic, biotic factors, exactly, yeah. So if we're going through this limiting factors, there are 2 ways that we can define limiting factors. There's abiotic, biotic, right? As you guys know, abiotic is non-living, so things like space, things like temperature, things like pH of the water if you look in aquatic environments. Biotic are things like interactions with living things, predation, things like that, competition, right? But we also got another way to identify limiting factors. Again, limiting factors are things that change the carrying capacity, right? So there's density dependent and density independent. I'm going to show you guys a trick today in terms of when a question asks you predict and a cause for a population having the decreased carrying capacity or increased carrying capacity, which one of these do you pick it from? So I'm going to show you. Essentially, density dependent is things that are affected by or are caused by having an increased number or decreased number of organisms within the population, right? So density dependent limiting factors involve things like competition, right? The more it is linked to how many organisms there are, the more organisms, the more competition, right? Maid availability, yeah, things like that. Maid availability, right? These are all density dependent. Even as well, you would even say things like disease, right? Because it's linked to how many people are in the population. Because if there are more people in the population, disease, yeah, you're good. I'm going to rejoin BRB. Yeah, you're good. Disease is going to be worse with more individuals there, right? Density independent are things that affect the population regardless of how many people are within it, right? So what are aspects of density independent? Can you guys rapid recall that one? Density independent. Natural disaster, yeah, you can't control it. Exactly. Natural disaster, that's a big one. That's a key one. Right? When in doubt, say natural disaster. Water availability, weather, perfect. Things like that. Space as well, like nesting space, like things like if there's a natural disaster that results in. So another key mistake that students make is when I'm going to give you guys a question and hopefully you guys can try this one out. So let's do this one here. This part. Question 5. The graph shows a change for a penguin population and the theoretical carrying capacity. Oh, I lost it. There we go. Pointing on the graph indicates a change in the carrying capacity triggered by. Okay, so we've got A, an increase in penguin pathogens. So this is pretty much saying what could explain a change in carrying capacity. And we've got lots of viable options, right? So what is it? What's the actual answer? Like, how do we know, right? We mentioned disease is a limiting factor. We said mates availability is a limiting factor. We said space is a limiting factor. And this seems correct as well, right? How do we actually know what the answer is? Can anyone give it a go? I'll get you guys to think about it first before I kind of explain the question and what's the trick here. What's the actual aspect here that we're trying to do? So some people saying C, wouldn't it be the factor with the quickest change? D, is a gradual decrease, high rate of change. Is it because the QCAA def says that resources and services in the definition for carrying capacity? Is it probably D because it's a reduction in carrying capacity, not population? Alright, so we've got varied answers. Mostly between C and D. So Tia said, is it because QCAA definition says resources and services in the definition? Kind of explain what you mean. I'm not sure what you mean by that. But I'm going to tell you guys essentially the key point for determining things that change the carrying capacity. So you guys you're technically correct, right? Carrying capacity, right? Carrying capacity is limiting factors, things that do change carrying capacity. But it's whether they change it indefinitely, that is linked to the density, or they change it that is density independent, is what we want to care about here. So if it's things like predation, let me just draw you guys a quick logistic curve, which is logistic means S shape, right? So it's reaches the carrying capacity. If at this point in time I were to increase the predators, right, what would happen? Can you guys tell me what would happen? So what is this again? This is a density dependent, because it's linked to the amount of people there. Yeah, you decrease the amount of population there because they're getting preyed on, right? However, this is a key point here. If we're looking at density dependent factors, this is a key thing that lots of students mess up, right? We're looking at density dependent factors. So this is the key. I want you guys to remember this. Density dependent factors do not change the carrying capacity indefinitely, because what will happen? You get a decrease in the prey, but what will happen? Because yes, even though there is an increase in predators, what will happen is that because there's a decrease in the organism they feed on, they will no longer be able to feed on them as much. So therefore, it will start to rise again. Does this make sense, guys? So density dependent factors affect the population in a very cyclic way about the carrying capacity. So imagine this is the carrying capacity. If there's density dependent factors, they will influence it to increase slightly or decrease slightly, but it won't actually indefinitely change the benchmark. It won't just move it down here. Yeah, exactly. Does this make sense, guys? This might be a complex concept for some people. Yeah, thanks Tia for putting that. That's awesome. Yeah. Does that make sense, guys? If we have density dependent factors, these are things that will oscillate the organism numbers. Right? They won't actually decrease them indefinitely or definitely, right? Why is it density dependent? Well, because predators, right? If you want to think about predators, right? The amount that it affects the population depends on how much is there, right? Whereas if you're looking at a natural disaster, that's going to affect them regardless, right? But if you have predators and the density is low, that's not going to make a difference. Does that make sense? So that's why it's density dependent, right? So the key point here, right, is that density independence, limiting factors, change the carrying capacity indefinitely. Key point. So when we look at if it's density dependent or independent, do we look at how quickly the curve has gone up and down? It's not really much to do with how quickly. It's more to do with, is it oscillating up and down, or is it just straight change the threshold, change the full carrying capacity for good? In this case, it's changed it. It's gone from here down to here. So it must have been a density independent factor, something that has occurred in this population. Now let's rule these things out. Increase in penguin pathogens. That would not decrease the carrying capacity the whole way down because it's linked to how many are there. So that's not it. A decrease in number of mates available. Again, right? This is density dependent, right? Do you guys agree? Right? Density dependent. So it won't be that one, right? A reduction in space available for nesting sites. Reduction of space. What could that be due to? Natural disasters, things like that. There's less space for them to nest and grow. Yes, this one will decrease the carrying capacity for good, right? And if you want to look at the last one, just for clarity's sake, because some of you guys said D, an introduced disease affecting the penguin survivability rates. They would survive for less, correct, but will that impact? Remember, the definition of carrying capacity is how much organisms can an environment withstand at a given period of time, right, for a species. So if there's more diseases, we'd probably kill off a few, 100%. But will that actually change the environment's ability to withstand the population? No. Does that make sense, guys? So that's why it's not D. So lack of space meaning geographical space. So that's a good point as well, Veronica, right? You said, could the lack of space be due to the large population? That will, an increased number of population, yes, will give less space for them to exist. You're right. But what this is referring to is the geographical space is cut down, not the relative space. But that's a good point, right? So this is saying a reduction in space available for nesting sites. This would be due to, this is something that will decrease the carrying capacity for good, because think about the definition, which is why I asked you guys carrying capacity definition, is the amount of organisms that a ecosystem can withstand, the maximum amount. So if we are decreasing the space for them to nest, that will decrease the amount that it can hold. If it was density dependent space reduction, would it say relative reduction of space? Yeah. So density dependent space reduction is like essentially like this is kind of getting a bit more on the complicated side, but they wouldn't ask you something that it would be pretty obvious, right? But you'd say like a cause for a carrying capacity to exist is when it's reached the space threshold. That could be a reason why the carrying capacities exist, or resources or things like that. So, okay, cool. So if we're looking at this question here, now to test this knowledge, right? Let's look at this question. Identify 2 modifications that could cause this change and explain your reasoning. Let's try and do this one. So, art of answering bio questions. Identify. This is a double whammy, right? We got identify and we got explain. Okay, right. Okay, and what is this? A 4 marker. Okay, so we got a 4 marker. So we got a 4 marker here. So what could happen here? 4 marker. So where are the 4 marks coming from? Let's dissect this. Identify 2 modifications and explain your reasoning. Well, because it's 4 marks, I'm assuming it's identify 1 reason why that would change the carrying capacity. Identify the second reason why would that change the carrying capacity. Let's do it. So first reason. What could be 1 reason? Exactly. Perfect, Nova. Density independent, because that's what we said, things that change the carrying capacity for good, right? Right, so this is just a case of, was the carrying capacity before, and something would have changed to move it up, right? We don't know where it is because the question hasn't shown us the plateau, but because it has gone from a plateau going up, we can tell there's been a factor that has changed that has moved the carrying capacity at a higher threshold. So things like, what do we say are density dependent? Density independent. Space, food availability, yeah, things like that. So the easiest 2 ones would be space and food, right? An increased number of resources, an increased number of space for them to replicate, right? Because we're looking at, it doesn't say what organism this is, but I'm assuming because of the number here, it's looking at bacteria because it's such a high number. Yeah, exactly right. So how would you explain these things? Okay, good point. So let's say we want to use space. So this is the key part that students miss. They can identify what would change it, but they struggle to explain. And because it's a learning goal, you have to be able to do that. So why would space increase the carrying capacity or increase the population? And the reason I'm asking you guys rather than telling you the answer is I feel like this is how you guys can learn and actually remember it, by trying yourself. Yep, so you guys want to be careful as well when saying nesting sites, because I think this is talking about bacteria, so that wouldn't really apply. But the whole concept is there is more area for organisms to replicate replicate, reproduce, and grow, right? Therefore, increasing the available, like the places they can be found, therefore increasing the population size, right? So there's more area for them to reproduce and grow, right? Whereas food resources or resources, food, things like that, why would that increase the population size? Yes, you do. Reason being, and this is why looking at you do have to explain each point, because what does the question say? Identify and explain your reasoning. So it's identify two modifications and explain your reasoning, and it's a 4-marker, right? So that means one modification, explain why. Second modification, explain why. 4 marks. That's why it's so important here. Use your art of answering bio questions. It's the cognitive verb and the mark counting, right? So the reason why food, just to keep it quick, guys, to increase population size, because we're increasing the resources that the population needs to sustain itself, and therefore the population can grow beyond its original carrying capacity, because there is an increased number of resources to sustain growth. Because there are 2 factors here that actually impact carrying capacity. There's 2 principles you want to refer to: fecundity and survivability. Right? So survivability has to increase, or fecundity has to increase for the carrying capacity to go up. So these are your fundamental reasons you want to go back to, right? And why do these increase? It'd be obviously the reasons we talked about, but then also it wouldn't hurt to use the word tolerance range. If something's within the tolerance range, it's also going to thrive more, and therefore survivability is going to be higher. But it comes down to these 2 reasons: survivability and fecundity, right? Fecundity, just to clarify, guys, it means how easily you can reproduce, right? So if that increases, you can reproduce more, and that would make sense for the carrying capacity to go up in that case. Okay. Does that make sense, everyone? Before I move on from this concept, it's something I really want to touch on because it's a classic question. It comes up again and again with QCAA. Every single time you're guaranteed a question like this, you have to understand these reasons. All right? Okay, sweet. Too easy. Okay, and then obviously this question is along those lines. I'm going to give you guys this resource so you guys can try this one in your own time. It's from QCAA, and this one as well. Same concept. All right. Let's go on to this here. So now, classification process. I did really want to touch on this stuff because, and I've titled this a funny title, but beware of the big dog. Because there's a big dog here. There is a big dog in this topic. Can you guys try and guess, out of these learning goals, which one is the big dog? Which is the one that is just, I don't even want, it's just stupid. This one right here. Well, I'm going to get you guys to guess first. Well, I already told you. I've already given away. But, yeah. Ratified sampling. This is the big dog of this topic. Classification processes. Look how big this learning goal is. Exactly, yeah. There's a question like this every year because it's so annoying. What is this? You guys are right. It's such an annoying dog. But I'm going to tell you guys how to actually make sure that you're covered for this, right, in a sec. So, what the hell is this talking about? Describe the process of stratified sampling in terms of and they go on and on and on. It's like, how the heck am I meant to answer this? This is a big dog. This is another big dog here on top of it as well. Explain how the process of classifying ecosystems is an important step towards effective ecosystem management. This is just bullshit as well, right? But I'm going to cover this one in another session. I'll cover this one in another session because we don't have time. But this is the one we're going to go through today. So, before I get into that big dog, I just want to ask you guys, what is the difference between a sampling method and a sampling technique? Because they're different things. Method is what you're doing and the technique is how. That's a really good point to do. Yeah, that's actually a really good description. Can we be more specific in terms of, can you guys give me examples of sampling techniques and then give me guys examples of sampling methods? So let's start with the methods first, because the methods is the umbrella broad thing. Exactly. Yeah. Random sampling. Random sampling. Systematic. Right? And then the big dog, we always don't want to forget about stratified. Stratified sampling. Right? And what's unique about stratified? Why are they hopping on? Why is there like a full learning goal about stratified? Why is there a full learning goal about this thing? Well, stratified, what's unique about it is it allows you to assess ecosystems along an environmental gradient. What the heck does that mean? I'll tell you guys in a sec. The ecosystem essentially, like, there's some ecosystems, there's different aspects of the ecosystem. Okay? So, like, if you're assessing mountain ranges, there are different elevations, for example. If you're assessing the sea, there are different depths at different areas. So the reason why we use stratified sampling is to get a picture of the ecosystem as it changes along its gradient, right? So this allows us to detect, like, look at the ecosystem as a whole and examine each strata relative to its contribution. A strata is essentially a piece of area from the ecosystem that is characterized by a feature. So, for example, if you're looking at depth level, this is like the sea, and it gets deeper and deeper and deeper. This is water, by the way. This thing is water, right? Gets deeper, deeper, and deeper. We can use stratified sampling to assess any dependent variable that we want to measure along the gradient, the environmental gradient, which is increasing depth of water. This is depth, right? So each strata, we separate it into strata. So let's say this is shallow. This is medium. This is deep. You guys get the picture, right? So it allows us to assess an environment over a changing environmental gradient and get a picture of the whole thing. That's why systematic sampling is probably the best one, and that's why there's a full learning goal on it. Okay? So it allows us to get a representative shot of what the sample is. Now, if we're looking at sampling techniques, these are how we actually measure the dependent variable of a sample. So we got sampling techniques such as, we got quadrats. You guys know what that is, right? We got a line transect. And the third one is a belt transect. And I did want to get into the difference between these two, but essentially a belt transect is the best of both worlds. You're getting a quadrat as well as a line transect. So essentially it's a line transect with quadrats placed at intervals throughout. So it makes like a belt. So you measure this, you measure this. So it's a quadrat coupled with a line transect. So it's like a quadrat and a line transect at a base, and this is a belt transect. When in doubt, this is the best method. This is the best technique, sorry. Okay? Because it uses the advantage of both. Okay? So this is something you guys want to consider as well. What are the pros and cons of each of these methods? Right? So it's quadrat here, line transect here, belt transect. So what's the advantages of using a quadrat? I'm going to go through real quick. Easy. It's easy to use, right? Cost effective. There's a dollar sign. Cost effective, right? What does it measure? What can it measure and what can it not measure? There's a con of it. What can it measure? Abundance? Can't measure distribution? It's actually the other way around. Quadrats measure the distribution. Can't measure the abundance. Key point. Why does it measure distribution? Well, if you want to think about a quadrat, you've got these grids within the grid, right? It's used to measure things like percentage frequency and percentage cover. So percentage cover is, for example, if I had like someone trying to measure here, this would be a percentage cover approximately 1 square, 2 squares, 3 squares, 2.5 squares. So it's a percentage cover out of 9 squares, 2.5 over 9, and you'd calculate that as a percentage, right? Right? So yeah, money saving. So you save money. Sorry about that. Yeah. And then you got percentage frequency, which is, what's the difference between these two? Percentage frequency is how many boxes, many grids, does it appear in? Whereas percentage cover is how much of the grid it covers. Right? Subtle difference. Subtle difference. Percentage frequency. What would be the percentage frequency of this example? It would be, okay, remember, it's defined by the amount of grids it appears in. So this is 1 grid, 2 grids, 3 grids out of 9. So it's 3 over 9. So therefore it's 33%. All right. Just to explain, guys, what I mean by cost effective is a quadrat is not costly to set up. It's quite cheap. So that is an advantage of using a quadrat rather than these other techniques. So that's what I meant by that one. Does that sort of make sense, guys? Yeah, okay, sweet. So it's easy to do, it's cost effective, it measures distribution, it does not measure abundance, and also, big thing here, it doesn't measure motile things, right? Things that move around. You can't measure a rabbit with a quadrat. Just fuck the quadrat over the rabbit, right? No, right? And you can't measure and high vegetation. Reason being, if you got a tree, you can't measure it with a quadrat. You can't do that, right? It's just not, it's more low-lying vegetation, right? Low-lying things. That's a limitation of a quadrat, right? Now, in terms of a transact, it's literally the opposite. We can measure abundance. We can't measure distribution. Reason being is because it doesn't give us an area. It just gives us a distance. So it's hard to measure distribution. Does that make sense? And then also, in terms of its other advantages, that's pretty much it. Keep it very simple, and that's it. And a belt transact is you can measure both. So this is why I'm saying the best of both worlds. If the question is asking you to identify a type, suggest a sampling technique. When in doubt, just choose belt transact. Right? So it measures both. Disadvantage of it is it can be time consuming, right? And complex. Yep. Okay. So without further ado, I'm going to get you guys to test your knowledge here. So now that you guys understand the techniques, also I do want to introduce another one, which is mark capture, recapture. And this is to do with the Lincoln index, which I will get to in another session, but it's that formula you guys heard where it's M times N, it's N times M over M to find the estimated number of population. And the key about this one is it's for mobile organisms, things like rabbits, right? So think mark capture, recapture is mobile organisms. Everything else is for immobile organisms. So let's test your knowledge here. Can we guys try question 2? Yeah, you should know why you're doing which one. So just to answer that question, if you're asking which technique, you have to know the advantages of each one, which is why we went through the pros and cons. You guys should be now able to explain which one you've gone for and why. So what are we saying? So we're going some of you guys B. I want you guys to all try and give me an answer for this. So read the question very carefully, guys. So we can rule out a few instantly, right? We can rule out mark capture, mark recapture. Right, we can rule out that one because we're using mark recapture to do eucalyptus trees. That's instantly wrong. A transact is used to determine the abundance of rabbits. We can't use that to do abundance. That's instantly wrong. Because between A and B. So a transect can be used to determine the distribution of eucalyptus trees, or mark recapture is used to determine the distribution of rabbits. Uh-oh. Nervous onto it. So a lot of you guys said B, but here's the trick. What do we say mark recapture can do? It's the number of population, the number of organisms. So what is this question saying? Distribution of rabbits. It's not the abundance of rabbits. Remember, abundance is the number, distribution is the percentage they are in space. How is the mark recapture meant to find distribution? It can't. Trick question. I got you guys. This is wrong, right? It finds the abundance of rabbits, not the distribution of rabbits. So our answer is A. Got you guys. All right. I got you, a lot of you guys with that one. But hopefully now we learned that. And that's the key part. We're getting things wrong. It's good because now we know why we got it wrong. Okay? All right. Cool. Now, going to the big dog. I do want to get to the big dog. So, and I have lots to cover. I actually had a lot planned today, but we will cover this in the next session. So do not worry. But how are we finding this so far? I can't tell. Are we sheer boredom? Are we like pure engagement right now? What level of engaged are we right now? I like it. How are we all feeling? Yeah, super engaged for real. Me too. Soaking up the knowledge like a sponge. We're feeling good. Yep, love heart. Awesome stuff. Good stuff, guys. I'm glad you're liking it. All right, let's keep going. So, the big dog. All right. So, every single year there is a variant of this learning goal. Okay? And I'm going to give you guys the one from 2022. Right? So, this is essentially they're talking about the depth of water. I'm going to get you guys to answer this one for homework, but I'm going to tell you guys how to actually answer. And they've got the little marking guide here in case you guys need it. To answer this question. But I'm going to show you guys how to actually address this whole learning goal top to bottom in terms of describe. Make this a bit bigger. Describe stratified sampling. So there are multiple aspects to this, and I'm just going to dissect each one, all right, together. So, yeah, some of you guys have done this in class. So I'm not going to waste, if some of you guys have already done it, I'm just going to show you guys how to actually answer these questions. So in terms of stratified sampling, to describe stratified sampling, there are multiple aspects to it. So this is the acronym I use, and I don't even know, like, you guys just have to remember this. PDSSMP. Okay? Make some acronym, chuck it in the chat GPT. You should chuck out a mnemonic for you, or you can just remember it. And this is what each of these mean. Purpose. So what's the purpose of systematic sampling? We just said that at the beginning. It's to look at different strata along environmental gradient. Okay? Look at different strata over environmental gradient. Distribution. Sorry, not distribution. What is the dependent variable? That's the second one. So what do we say dependent? What we're measuring. What are things that we can measure? Right? So if we're looking at this question here, let's try and answer this. So the purpose of systematic sampling. We are trying to measure, in this case, the purpose is we are looking at how an ecosystem recovers after a widespread loss of seagrass from 4 to 60 months. And we're looking at how it recovers over time. So not only are we looking at a spatial scale, but also a temporal scale. Remember, spatial means time. Temporal means time. I'll correct that in a second. Temporal means time. Spatial obviously means along a space, right? So spatial, we're looking at like across a distance. Temporal is across a time. The time is from 4 to 60 months. Right, and the space is we're looking at how it changed. Remember I said the purpose of stratified sampling is to look at ecosystem profile over different environmental gradients, or like looking at environmental gradient. So in terms of how we're going to set this up, we have to explain to QCAA because it's asked us to describe how stratified sampling could be used. So we just said that's the purpose aspect. right? So we're going to explain that stratified sampling is to look at the recovery of seagrass from 4 to 60 months by examining its growth over a spatial scale, and examine where are we actually going to put this, where are we actually going to measure? Well, it has to get all the strata, right? So it would not make sense to have our data set up like this, because we're not getting all the layers. Remember, stratified sampling is getting all the strata. So you would explain, it would involve setting up, what does this look like? Is this a quadrat? Is this a quadrat, a line transect? And is it more transect or a quadrat, what I've just drawn? Transect, yeah, exactly. So what we're doing here is we're looking at it over a transect. This could be line or belt, but remember how I said, when in doubt, choose belt because it's better. It just does the best of both worlds. So yeah, could do a belt transect, and this is looking at north, approximately how long is this? 5 kilometers north. So we've got 5 kilometers north, and that's the first part. Describe how systematic sampling could be used, right? And then identifying. So that's the purpose. Dependent variable is what we're going to measure. So what can we measure with systematic sampling? We can measure anything. We can measure percentage cover, percentage frequency. If we're looking at seagrass here, abundance wouldn't really be a big one because we can't count every grain of, or every strain of seagrass. It would be mainly 4 marker. So that's the art of answering bio questions. I was, because we're running out of time, I didn't go through this, but 4 marks. So that means you're going to look at, you're going to ask for 4 things. Let's try and identify what 4 things they need from this question. Let's do that. So describe how stratified sampling can be used. What do we say describe was? This was looking at, like, to say what's going on and how it could be used. So this is a 1 mark. We just said that. It's going to be used to look at the recovery of seagrass from 4 to 6 months. Right? How? So how are we going to do that? We're going to set up a quadrat north, which is around 5, let's make it like on the higher side, 6 kilometers. And it's going to be measuring it. If we're going from 4 to 6 months, we can say we're going to measure it monthly. Right? So see how very specific terms is going to get you guys the marks. So that'll be another mark. Right? So the purpose and then why. And then what's the second part of the question? Identify a survey technique. Identify, what does that mean? It's just state it. So easy 1 marker. Remember how we said identify 1 mark. So it's all coming together, guys. Right? And then what's this, the 4th mark? The purpose of the study. So this will be examine how it would recover over a temporal scale, all the strata. What would the surveying technique? All right, what do we say? We can justify 2 of the 3 surveying techniques. So the first one we can't justify. Quadrat, you would not use a quadrat because we're looking over a longer distance. So you can use a line transect or you can use a belt transect. Belt transect is just the best of both worlds. Remember, guys, when in doubt, belt transect. Okay. So, and then how we can do it. So let's kind of recap, guys. So we've identified our 4 marks. Let me recap this for you guys. How stratified sampling can be used. So we can say, like, what are we going to measure? So dependent variable, that'll be a 1 mark. So what could we measure? We can measure percentage cover. We've identified percentage cover. Boom, that's 1 mark. Second mark could be we'll measure monthly, and we're going to set up the quadrat 6 kilometers north to get all the strata off location A. That's the second mark. So we say how. Right? Because remember, it's how and describe. Right? Third mark, identify a sampling technique. We'll use a belt transect. And then the purpose of the study is to measure the recovery of the seagrass over the 4 to 60 months. Boom. We've answered this question. And this is actually the hardest question in 2022. All my friends spazzed on it. But because I actually looked at the learning goal and understood when I actually answered this question in 2022, because this was my exam, all I did was spam through this stuff and I got 4 marks. So I didn't know sometimes what the question was asking. So I just went through all of them and I got 4 marks. So let me go through the rest of them. So we've gone purpose, we've gone dependent variable, The second S is going to be the site selection. So what does this mean? This is what we've talked about. How are you going to set up? Where are you going to do it? Where are you sampling? And why? So what do we say here? We are sampling 6 kilometers north of location A to get all the strata. Second S, surveying technique, right? That question already asked for that. So we already got that one down packed. And then what's this one? Minimizing bias. So I did this in the question as well. And I think you could have got a mark off that alternatively as well. So minimizing bias. What does that mean? What are ways we can minimize bias? Tell me as much as you guys can in the chat. Just spam me in the chat. What are ways we can minimize bias? Let's go. Counting criteria, sample size. Yeah, we increase sample size. Be specific, guys. So increase the sample size. Absolutely. Calibrating equipment, number of generators, also random number generator. Perfect. So we can randomize it with a number generator. Perfect. Yeah, good stuff, guys. Awesome. Calibrate. Awesome. Well done. Now, and then what's the last one? This is a bit niche, but it'd be a good one to add as well. Presentation. So what this means is, how are we displaying the data? Is it a graph? Is it a ecosystem profile? Like, how are we going to do it? Are we going to do it like this? Are we going to like visually, diagrammatically show the depth as it changes? How are we displaying the data? It could be a table, right? How are we displaying the data? Does that make sense, guys? I've just dissected the big dog of the learning goals, which is this one. So hopefully you guys understand what they're actually asking and how to actually answer questions related to this. Can we get a thumbs up for this before I move on? How are we all feeling in that big dog? Are we feeling more confident to ace that big dog? Yeah, obviously we have a lot more to cover. But the exact theories that I'm teaching guys, notice that it's not just concepts, it's how to actually answer the learning goal. How to actually the learning goals, and then also the common questions that we get related to those learning goals. Okay, so not 100%. What does it mean by calibrate? So what I mean by calibrate is a way we can minimize bias is to make sure that our equipment are accurate. So it's just making sure our equipment are accurate. So that's calibrating. It's a very niche word. It's not in the learning goals. So it's just one reason we can minimize bias. What do you state with the purpose again? So the purpose is we're going to measure changing environment over a gradient and different strata. So that's the unique thing about stratified sampling. It's over different strata. Okay. All right, cool. So, all right, nice. So yeah, notice, we're going to do a lot more in this session because I want to go back to understand the scope. We have already covered what? 1, 2, barely 3 of these dot points. So got a lot more to do, and this is what I plan to do for the rest of the sessions. Okay. So let's go through, let me do a bit of a vote because I've got 2 concepts left. The next one we'll cover in the second session. But do we want to go through these set of learning goals, biotechnology, right? Biotech made easy. Or do you want me to refresh protein synthesis along with mutations? Let's put a vote in the chat. Protein. Okay. Biotech, protein. Okay, it's okay. I think protein's winning. Unless more people say biotech, mixed opinions. Okay, this is actually very mixed. Okay, I'm gonna go through biotech. We got protein. Okay, we'll cover this in the next session, so make sure to stay tuned for that. All right, but I'm gonna go through biotech today just because it will allow me to get done on time. But we'll cover more of protein in the one more person says protein. That we'll do it next week. Sorry, not next week. When on week 1, the start of the holidays, and I'll get into that a bit towards the end. So, stay around to the end, guys, and also so you guys can get these resources as well. So, let's go into biotech real quick. So, if we're looking at this stuff, right? Why does QCAA teach this to start off with. What the heck is biotechnology? Like, it just seems so random. It's essentially just applications of what do you guys learn in terms of the DNA Unit 4 stuff. It's just applications of it, right? Also as well, are there going to be more sessions on the holidays per week for different subjects? Yes, absolutely. So I'll dive into that a bit at the end of the session. So these are the learning goals they put in for biotech. So I've just kind of recognized that's easy, right? Explain, that's a bit of the hard one here, PCR and gel lecture, and the medium one is describe the process of making recombinant DNA. But I just want to touch on this one. Appraise data. This is the first time I've ever seen this. Appraise. Like, what the F is this, right? Essentially, just to tell you guys, appraise just means to judge something. So if they were, and I haven't seen a question about this, but if they ever were to give you a question that says appraise, or like that targets this learning goal, it would need context. So this isn't something you can really learn. It's more like understand what biotech does, and you'd be given a context to answer a learning goal like this one. Very rare. I haven't seen it in much exam questions, if any at all. All right. So now, just so we get the learning goals straight, this is saying describe the process of making recombinant DNA and explain the purpose of PCR. So you wouldn't have to. The steps of PCR is not a learning goal. You don't need to know this. You just need to understand the purpose of PCR. And I'll tell you guys right now, the purpose of PCR is very simple. It is to so essentially you have genes, which are quite, like, obviously this extensive. You are taking a snippet of a gene fragment, a DNA fragment or segment, and you're zooming up on it. Zooming up by amplifying it. You are amplifying it so you can examine it. And how do we do that? It's polymerase chain reaction. So we are replicating it. We are replicating. So you end up with millions of gene fragments, and therefore you'd be able to, because you've amplified it, you can now look at it in detail, right? That's the purpose of PCR. Okay? So essentially, to give you guys a couple questions related to this. Explain the purpose of gel electrophoresis within DNA profiling. So essentially, PCR, one of the aspects of PCR is gel electrophoresis. And what is gel electrophoresis? So once we've done PCR on something, essentially we have taken a fragment of the DNA and we've zoomed up on it. A way that you can compare DNA fragments is through gel electrophoresis. And essentially what you're doing is you're placing the fragment on a gel slide, and you are separating the base pairs based off their size. So the smaller base pairs move further along, and the larger base pairs move further behind. So essentially, all you need to know for gel electrophoresis is you're separating them. Separating based off size. Alright? So don't need to go into any more detail than this. Right? So gel electrophoresis, it's an application of PCR. So after you've done PCR, you can undergo gel electrophoresis, which you're separating the base pairs, because within the DNA, you've got different base pairs, and you're separating them based off size to create a banding structure, right? Okay, so that's pretty much it. So that's this learning goal. Explain the purpose of PCR, right? And also, if we're going just to answer you guys this question here, recognize the applications of DNA sequencing. So this is kind of like, once you know them, you know them. But just to test your knowledge on this, to make sure how we're all doing on this, let's try question 9. Let's try question 9. What is the biotech application of DNA profiling? Which is, by the way, another application of PCR. So this is what we're doing. PCR is just used to amplify DNA so we can examine it. So that could be through gel electrophoresis, that could be through DNA profiling. What did I answer here? We're saying D, epigenetic analysis. The tempting option. A, gene therapy. Yeah. So we've got lots of contrasting opinions. Anyone else want to contribute here? Okay. B. All right. Okay. So essentially, to answer this question here, all you guys got to know applications of DNA profiling. Do you guys remember the shows like when you're scrolling through your reels and there's like shows that say like you are not the father and the guys go crazy, right? That's an application of DNA profiling. Paternity testing. So essentially, you're comparing. So think you are not the father, right? These are applications of DNA profiling. So things like paternity testing. Like, you know, the criminal, how they look at the fingerprint? Criminology? Forensics, exactly. That's the word for it. Forensics. Yeah, exactly. Another one is to test genetic security of foods, right? So remember, when we're looking at DNA profiling, we're zooming in on a gene, a DNA fragment to examine it, right? To examine it. So based off that, let's kind of rule out these options here. Gene therapy. If you guys remember, if some of you guys have done student experiments on gene therapy, but essentially gene therapy is we are looking at inserting a gene into a host, which can be a human cell, for a therapeutic effect. So this is not actually examining DNA in detail, its components. So it's not gene therapy. Okay. Drug discovery. Drug discovery is a weird one. It's like we usually don't use PCR to discover drugs. This is a different process. So there's nothing about examining something closely, right? No. So PCR is generally not used for gene therapy. And the reason being is because gene therapy is just you're inserting a gene into a host through a vector. Nothing to do with examining it. Yes, recombinant DNA. So that's a different concept. It uses recombinant DNA. So it doesn't use PCR, because PCR, essentially, when we go back to the beginning, when I guys told you, PCR is just zooming up on a DNA fragment to examine it in more detail, right? Recombinant DNA is different, and I'll go through the steps in a second because that is a learning goal. Describe the process of making recombinant. So I'll go through that in 1 second. So PCR, all you need to know is the purpose and then its applications. So food drug discovery, we don't. Epigenetic analysis. Okay, let's think about this. What is epigenetics? Epigenetics is factors that regulate your expression of genes. So that has nothing really to do with amplifying DNA itself. This regulates the expression of DNA, the proteins that change the expression of DNA, right? So it has nothing to do with amplifying a DNA and examining it. So that's actually also not an application of DNA profile. So because I told you guys one of the reasons is to check security of foods and make sure that the DNA within foods is desired, that is the correct option. The answer is B here. This is a tricky one. I'm not gonna lie, this is a tricky one. But once you know the applications, you got it. So this is more like it's easy because once you know them, you're locked in. Right? You can easily tick this learning goal off once you know them. So here they are here. Right? Paternity testing, things like that. You can also meet like ancestry tree. Yeah. Comparing and analyzing DNA fragments in detail. That is the purpose of PCR. Right? You're taking a small fragment, you're duplicating it a bunch of times so you can actually examine it. Okay? Does that make sense, everyone? Before I get to recombinant DNA, before we finish up, does that make sense? Is everyone all good on that? Thumbs up. Yeah. So you're taking a subsection of the DNA strand, a segment. You are replicating that a bunch of times, so therefore you can examine it. That's it. Simple as that. So you're not duplicating the whole DNA fragment. You're only taking a small segment of it, not the whole strand. Yeah. So it allows us to look at genes in detail. Okay. So let's go through recombinant DNA just very quickly. So the purpose of recombinant DNA is different to PCR. So you got different. These are different. Making recombinant DNA. The purpose of that, right? The purpose of making recombinant DNA is to deliver genes or like desired genes to a host. Application of that can be gene therapy. Right? We're trying to deliver desired gene to a host. That's the purpose of recombinant DNA. Okay, so we are making DNA. Yeah, CRISPR-Cas. Yeah, correct. All right, so recombinant DNA. Let's go through the steps because that's the whole learning goal. So once we know this, we can tick it off. Right, so I just want to focus on learning goals here. Right, so step 1, isolation of DNA. What are we doing here? This is simply, we got a full strand, right? But we only want to give a host the desired gene. So what do? We take off a segment out. So we isolate the DNA fragment that we want. Does that make sense, guys? That's isolated DNA. All right. Okay, let me ask you a question. What is the little enzyme, the cheeky buggers that snip this thing off? You guys have to know this. This is a key point. Restriction enzyme. Exactly. Good stuff. Bit of rapid recall here. Restriction enzymes. Right. Think scissors restriction. Okay. So that's isolated DNA. What's the second part? What's the second aspect? What are we doing? Insertion. Insertion of DNA. So what does that mean, right? What we're doing is we are combining, or we are putting the DNA, just before I get to this also, I just wanted to really highlight for restriction enzymes. They create certain ends when they snip the things off, and there's 2 variants of this. So essentially what we're doing, we're snipping this part out. And there are 2 types of ends. There's something called sticky ends. So what they create is, because remember, this is a DNA fragment which is double-stranded. So remember how you have codons, and then you have complementary, right? You got the complementary codons. So these are 2 here. But when the restriction enzyme actually snip it off, it creates this like this step thing. It doesn't snip it off straight like this. It snips it off so one, it's in this way, and then the other one is snipped off like this. So think about like Legos, right? It's snipped off in a way. Imagine like you got a Lego brick, and these are like the little ends on the Lego brick. It's snipped off like that, not like this. Okay? Does that make sense? So these are called sticky ends. Sticky ends. Does that make sense, guys? So that's an important concept to recognize. When the restriction enzymes snip and isolate a DNA fragment, they create sticky ends. So technically, it should look a bit like this. All right. Is this related? Is this about 5 and 3 ends, or are they different? So 5' and 3' is just the direction that they go, and they're anti-parallel, so they go like this and this. I will get to 5', 3' in the next session when we talk about protein synthesis and replication, things like that. I will go in that in lots of detail. So yeah, don't worry. So sticky ends. Does that make sense? So then, insert the DNA. So what happens? We put in our DNA fragment with a sticky end. It looks something like this, right? And we insert it, and notice the reason, and this is coming back now to the reason, the significance of this sticky end, is that now it can join beautifully with our vector. What is a vector? This is called a plasmid. And this is just like think of the plasmid as like the limousine, the limo. So this is just a transporting the actual desired DNA fragment into the host. It's a way to actually transport it. Yes, exactly. And it inserts, right? The third step is joining of DNA. And as you guys guessed it, this is the joining, where they join to form this is called a plasmid vector. So it's a transport aspect with the plasmid. Make sense? So this is where our DNA fragment is now. Let me highlight that in a color so you guys know this is the fragment that we wanted to transport in the first place. It's this one here. All right. So this is to create a plasmid vector. Now, another key one. So we've mentioned one key big dog here, which is the restriction enzyme. There's another one that actually plays here. And this one, right, this enzyme is responsible for gluing the DNA fragment to the plasmid to create the vector. What is this one called, guys? Ligase. Awesome. DNA ligase. Good stuff, right? We are ligating, essentially gluing the DNA fragment into the plasmid to form a plasmid vector. The last part, and this is the part that confuses some people because they don't know the point of it, but we got the plasmid vector here. So I'm going to draw this plasmid vector. We are going to put it, this is called amplification. I'll tell you why it's called amplification in a second. So we are going to put this plasmid vector into a bacteria. This is a host. And why the heck have we decided to do that? You guys try and know what's the point of doing that? Replicate, exactly. What are bacteria really good at? Replicating, right? So we are putting this plasmid vector into the bacteria to duplicate its numbers, to actually replicate it, because bacteria, why we actually use bacteria is number one, it can very, very fast replication. So this allows us to actually amplify, this is why it's called amplification, to amplify our recombinant DNA, right? They're really good at replicating, and also another good aspect of it is it's ethically good. It's ethical, right? We can't use human cells, for example, right? So it bypasses the ethical consideration. So it's like ethical to use bacteria rather than human cells, right? So that's also a great aspect as well. So they could ask why the host bacteria, that's an easy question, but they could technically ask that, and you would say because it has very high replication, it's ethical, okay? And it's good at actually hosting different genes that you put into it. All right. So those are the 4 steps of recombinant DNA, making recombinant DNA. Does that make sense, everyone? That's it. That's all you need to know for that learning goal. And that pretty much concludes the end of this session. But obviously, stay around. We're going to do a little announcement that will allow you guys to receive these resources as well. But how are you all feeling? I just want to ask, because I asked you guys all at the beginning how we felt for bio externals. After this session, can you all guys rate 1 to 10 felt for this one here? How do we feel after this session? Everyone drop what they think. Much better. 7, 8, 7, 7. Awesome. That's great, because assuming I was we had lots of 2s and 3s at the beginning. Awesome. Yeah. Everyone drop what they're thinking right now. How are we fine today? Uncooked. I reversed your cooking. Yeah, good stuff. Awesome. Good stuff, guys. Yeah, great. I'm glad you guys liked it. And yeah, feeling good. Awesome. This is beautiful stuff. All right. And would we who here would attend another one of these sessions? Because obviously, if I look at understand the scope, I could only run through too much today. Who would come to another one of these? Me. Awesome. Amazing. Yeah, good stuff. So just to go a bit back on what we'll be able to do with these students as well. I've dived into the results we'll be able to get, and hopefully you guys can see some of these students in here as well. But I just want to touch students that came to our masterclass. They're able to get 100. I love that gift. Awesome stuff. We've got students that came back with 100% on their specialist exams, which is crazy. We've got students that come back with like 20 out of 20. We have students currently with us this year that have gotten 20 out of 20s on all their assignments so far, and they're currently in these workshops as well. So really, really excited to go through and the results. And do we feel like the 90 minutes paid off? Absolutely. So I'm glad to hear that. If having even learned the content, what would you say is the best way to study? Number one, I told you guys, everyone should do this after this session. Highlight red, yellow, green traffic light method down this learning goals. So look at all the learning goals, and I'll give you guys this towards the end. So just stay around. Just thanks for your patience, guys. Stay around a couple of minutes, and you guys, I want you guys to highlight what you feel confident in, what you don't feel confident in, and if it's mostly red, you got some work to do. So that will give you a good starting point. But the motive of these sessions and the reason why, let me just share my face so you guys can see me. Yeah. So the whole purpose of these sessions, and I'll run top to bottom all the learning goals. But notice, I'm not just running through the content. I'm running through what the learning goal says and where and how that is assessed in questions. So it's question exposure and teaching you guys the content that quiz should have like specific looks for. And now keep building on the art of answering bio questions as we keep doing more and more. So the goal is, like people that came to me, and if like students like Thomas who came to me last year with 40% on their mock exam, and I was able to bump up to 80%. I'm sure for all of you guys, we can get outstanding results, and I will make sure that we cover all these learning goals, top to bottom. I've done a few today, and I strive to do the rest of them for you guys, leading up to the externals. And just to kind of, so could someone please text the link? That's the QR code. Yeah. So I'll give you guys that in a second. So, all right. So if you guys can please, everyone who came today should be filling this out. Number one, it's going to give us opportunity to get some feedback from you guys. Also, if you want to receive the resources, you have to fill this one out. And just towards the end as well, I did want to touch on our the future sessions and how they'll be running. So it will be starting from week one of holidays, and it will go all the way to your external exam. Every single week, 1.5 hours sessions. And not only will these be live, but I'll also record them as well. But the key thing here, it will be exclusive to students that sign up today, because we only want to work with students who are committed. Like you guys are joining on a Saturday morning. I love that. I want to help students like you guys out. So it's going to be exclusive to students that sign up with us. And not everyone will get access to these sessions or the recordings, or the live help we'll be giving you guys on the chat 24/7, because I know that obviously to get the results that I've been able to get for all our students last year and been working with the students this year, it comes down to exposing you guys to exam questions that come up again and again, but also giving you guys live feedback when you guys need. And that's something I wish I had. So that's why I will be doing that one for you guys, but only for the students that sign up. So this will be exclusive. This won't be on the Queensland Study Hub. This will be a separate forum within the Study Hub for people that only sign up. Okay. So sign up. Express your interest to sign up today by signing the QR code. Is there another link than the barcode? Ollie should be sending that in the chat. Can you guys hear me? Someone just said, wait, I didn't hear. Oh, exclusive, yeah. So essentially, this is only for people that sign up, and it's not for other students. So essentially, if you want to check out, if you press the QR code and you are interested, it will direct you to the site where you guys can see that it's for the lowest early bird price that will start on week one of the program. So this is what you call by the mentoring program. So this is what I'm talking about, the weekly sessions that will be 1.5 hours every single week, exactly like we did today, up until your external exam, and with live help on the chat if you guys need any help 24/7. So if you guys are interested in that, if you guys like today's session, we got a lot more planned to cover the rest of the learning goals. And as you guys can see, if you guys have more masterclasses that you guys want to sign up after today, so let's say anyone that's attending chemistry tomorrow, anyone that's planning to attend English next week, you guys can make that decision on how many subjects you want to sign up for the period moving forward up until your external exam. So we'll be doing this for all the core subjects: chemistry, biology, English, general maths, math methods, and physics. And it's cheaper as you enroll in more subjects. So if you guys are thinking that you might need help with more than just bio, you guys can enroll in more than one, and it is cheaper as you go. So if we are just including just the sessions that we run through, it is at a rate of $23 a session for one subject, $23 an hour, sorry, for one subject, and $20 an hour for 2 to 3 subjects, and even down to $18 an hour for more than 3 subjects. So if you guys want to think of this as a fraction of private tuition, and we're giving you guys this 24/7 chat help and an hour and a half workshops every single week with the resources that we have with Queensland Study Hub. So if you guys ask me, that's a bang for your buck right there. And this is not going to be the price that will last for the extended period of time. So if you guys want to claim this early bird price, as we like to call it, and if you guys have other students that are committed like yourself, feel free to refer them to this. And they can also claim this price, but this will be taken down as soon as we start our program. So anyone that wants to start after will be paying more. So if you guys want to sign up now, you guys are committed students. Again, we're running only a limited number of classes. I'm probably only running max 1 to 2 classes, which will be a limited number of people each class, just so I can give you guys more attention, help. Obviously today we had 30 people join, but during these classes, I'm probably going to do it only 10 to 15. So then that way, all your questions can be answered, and we can make sure that everyone gets the personalized help that they need to get literally the best that you can for bio, but not just that, also your whole ATAR, and get into the uni degree that you guys want to get into. So that's why we're only targeting specific students who are committed. And given that you guys joined today on a weekend, I'm sure that is the vast majority of you guys. So yeah. Has anyone gotten 50 for bio external? Yeah, even including myself, I got 49. I think it was 1 mark, so that I lost the whole thing. So I got 49 out of 50. 99 out of 100 for my bio. So you guys are in good hands. I've been teaching for 2 years. I just want to see every single one of you guys succeed. So that's why I am dedicated to making sure all of you guys get across the line. And as you guys can tell, with every single learning goal, there is a strategy around this learning goal, and there are common questions that come up again and again. And I'm not going to hold back on the value I give. So if you aren't signing up, you really are missing out. There's no other way to say it. And if you guys have other friends that will be committed, feel free to refer them the sign up link, even though they haven't attended today. And I'd be more than happy to send them what they missed today in terms of the recording, and then also give them the early bird discount if you guys are committed. So yeah. Can I get a thumbs up on the chat to those that have signed up or attended on signed up with the link below? Thumbs up, yep. Yep, awesome. Yep, everyone drop a thumbs up if you guys sign up, whether that's saying yes to our program or not. Just drop a thumbs up if you filled that form out, because we'd really appreciate it. Yep, awesome, thumbs up, yep. Cool, thank you so much. So the form, so just that form, yeah. So I'll pull it up again. Ollie, if you want to put it in the chat as well, again, just so students can see it. Yep, so it's in that forum. Sign up through that. And I look forward to seeing you in the future sessions as well. And also, just to touch more on a bit about what we'll be able to do for students in addition to our sessions. So if I want to look here, just in terms of these reviews, my confidence in methods literally skyrocketed. So this is referring to our masterclasses there. We've got students who have attended and were so much more confident, students with 20 out of 20 on all their assignments, students who joined, look at this, 15 out of 15 for specialists with our masterclass. Absolutely crazy. And hopefully you guys can see yourself as some of these students, because if they can do it. And if I can do it, you guys can too. I believe in you guys. So I'm actually fully invested to making sure that everyone gets the goals that they need for ATAR. And look at this, 2 weeks of joining. Significantly improved my assignment just from the IO3 workshop, spotted the weaknesses and were able to fix them. And we plan to do the same thing for these external exams as well. And then if you guys have the time to read the one on the right as well, QC Mentor has been the best decision of my life. Absolutely outstanding. So it's reviews like these that really keep us going and make sure that we want to do the best that we can. And this is kind of why the workshop, the program is priced as low as it is because that really, it's not really about the money for us. It's more about helping as much students as possible and giving you guys the support that I wish I had when I was doing the same journey as you guys. So really, 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 if you guys are committed students, like I was, press the sign up and then we will reach out to you guys further. So yeah. If you guys have any questions, feel free to put in the chat as well. But when do we have to sign up? So you guys can sign up anytime. And then also as well, if you want to increase the number of classes you want to do or decrease the number of classes you want to do at any period of time, there's 100% flexibility on you guys being able to do that. And on top of that as well, if you guys want to cancel for any reason, whether it's an emergency or something, we have no policy against that as well. So it's cancel anytime. It's really just for you guys. So you guys can do it for as long as you see the value in it. And I'm sure you'll see the value in it all the way through because I'll be dropping value bombs every single week. So if you guys like today, you're going to love what I got planned for you guys to make sure all of us max out our marks and max out our ATARs. So there are a limited number of classes we are running. So you ask, how do we know that if we are in the class so we can pay? Well, that's why I'm giving you guys the opportunity to sign up now, because obviously people that sign up now are the pre-order. So you guys will guarantee a spot in those classes in relation to students that sign up later for a higher price, but also not guaranteed entry. So obviously we will only charge you guys if you're in those classes. We're not going to charge you guys if you aren't accepted in those classes. So yeah. Do I just teach bio? Currently I just teach bio. This is just year 12 content. However, Katie, if you want to just reach out to me personally, and if you're in grade 11, just let me know and then I can sort something out for you there. But for currently these masterclass, these classes, just for the external exam sessions. So I'm running bio myself. There will be other tutors running methods, as you guys have seen Andy, if you guys have attended methods. And then also we got Bronte, who's a adjusted ATAR of 99.95, who's running English and running general maths. So you guys are in good hands either way. But as much as I really do like a lot of you guys, I feel like I'd get along with all of you guys. So I'm so excited to help you guys out in the bio class. So I'm only running bio, but there'll be other really good tutors running the rest of the subjects as well. So essentially I won't run along it in chronological order. I'll run through the high yield stuff because I don't want to teach from top to bottom. There are easy stuff, there are hard stuff. I'm going to cover the stuff that students miss, right? I'm not going to teach you guys as if the teacher would teach you guys. I will teach you guys as a student who's done well in QCAA, knows what QCAA is looking for, to make sure that you are exposed to the high yield, common mistakes and common questions that come up again and again. So you aren't surprised when you hit the external exam. You are ready for it. So I will try and cover the vast majority of the dot points. All the dot points that you're probably struggling with, I will cover those. Yeah. There's some easy ones that aren't worth covering, but the ones that come up again and again and are usually difficult from students based on my experience, I'll be covering all those. In addition to putting questions out that relate to those, not just actually covering the content. As you guys have seen through this lesson, there's an art to answering bio questions. So you guys will get the hang of it as we keep going. So what type of resources do we have? So we do have a learning goal resource. So it has learning goals in addition to questions relating to those learning goals, which I'll provide with you guys. And then also we are working on doing a content database where we collate all the content, as well as the notes that I give, the recordings that I give. And then in addition to that, it will have like extra exams outside QCAA that are on the Queensland Study Hub. But the key thing you guys get is feedback. So not only are you guys getting the exclusive access to the classes and the recording, so you guys can watch that anytime. If you want to go back to what I said back in like the first week, you guys can do that. But in addition to that, you guys will be getting a live feedback in the call. So if you guys have any questions about anything, throughout the day, around the clock, 24/7, drop it in the chat, and I'll be answering your questions throughout externals. This is something I wish I had when I was leading back, because I used to try and email my teachers. They never responded. So you guys have access to 99 plus ATAR students at the tip of your fingers, essentially. 100 times better than Study Insights. You're right, bro. 10 times better than Study Insights. Yeah, exactly. Exam Insights. Yeah. What about outside the calls? Yeah, so within the calls, you guys get me. And then outside the calls, you guys get that live 24/7 chat support by me as well. And I did forget to mention one thing as well. You also be getting accountability checks by me as well. So I will make sure you're accountable every single day, because I know a lot of you guys procrastinate. We don't want that to happen. So I'll keep you guys accountable and do these checks every day, saying, lock in, guys, give you guys strategies to focus, strategies to study, all that stuff. You guys can find everything in what we're going to give you guys. I'm so excited to work with you guys for the future. So everyone, if you guys haven't already, sign up, press the button below. Whether you are interested in signing up now to guarantee that spot, whether you need to refer some more friends, whether you want to think on it, just press the link below, or not the link below, but the QR code if you haven't already. And we'd love to see you on the other side. I'm so excited to do this. All right. Yeah, Ollie, was there anything else you wanted to add?
Oh, I think that's everything. Thanks for coming along, guys. We had a great turnout today. For methods, we had like 60, 70 people, so that was a really great class. But we got a good amount of people in this class. I feel like it was easy enough to answer every question, and it was just great to see the amount of engagement you guys were having during today's session. I was just watching the whole time, and it worked really well. So get ready for chemistry tomorrow, and get ready for general English next week.
You guys also saying 24/7, including 2 a.m. All right, 2 a.m. You guys might get me there, but if it's any waking hour, I will try and respond within the day. We'll do that. So
also, just remember, guys, if
no
you're
worries,
looking at getting into med, Jad is running JCU mentor sessions, so he's going to send an announcement out in the Masterclass announcements channel if you're looking for some advice about med entry. So listen out for that.
Yeah, so just I do JCU application stuff, but that's a side thing. Let's not put that at the center spot. We want to focus on you guys and making sure bio is stacked. Bio is not the one you want to be. Yeah, it's nice. Bio is not the subject you guys want to be worrying about with me. So you guys will ace that, so you guys have time to focus on everything else as well. Yeah, so my library is closing just in time, conveniently. So it was some grumpy librarian, bro. Yeah, all right. No worries, guys. Have a good one. Have a good rest of your day. And yeah, see you guys. Thank you, guys. Thanks for joining.